The first 8 chapters in the text are really a way of introduction, or rather than introduction, a foundation that we're going to take that information now and carry it through the rest of the systems and regions. So we're going to start into chapter 10. Chapter 10 and 11 are the somatosensory system, chapter 10 being the peripheral end of it, and chapter 11 being the central end. A lot of the systems in the book, they will divide it into peripheral and central. The peripheral nervous system is really anything that's not encased in bone. So the central nervous system is anything that is encased in bone. And so even though they're connected and one doesn't work without the other, we separate it just to discuss it and boil things down a little bit. So the rest of the chapters that we talk about in the book, which is all of them, except maybe chapter 23, but we are going to be talking about individual systems or regions. So there's going to be overlap between the different things. We're talking about the peripheral somatosensory system and the central somatosensory system in chapter 11. And then we're going to talk more about it when we talk about the cerebrum, because the central system includes parts of the cerebrum. So the information in chapter 11, I'm going to divide it up between 2 modules. The first part is the central somatosensory system. The second part is pain theory. And so the second part of chapter 11 and chapter 12 are all about pain. And so we're going to delve into it much more deeply than we have in previous classes. And that's a good thing, because a lot of what we address in physical therapy has to do with pain, or pain is the complaint that brings people in, or it's their entry to the medical system, and a lot of times it's their entry to physical therapy. So we want to understand well, we're not going to understand everything going on with pain, but we want to understand as much as we can so we can help our patients. Very important. So in this section, the learning objectives, and this is going to be divided between this lecture and the next lecture, but I want you to be able to define some terminology: stereognosis, a dermatome, which we've talked about previously, and a tract. What is the primary function of the somatosensory system? And at this point, you can go out on a limb and say, hey, is it somatosensation? And the answer would be yes. So I want you to be able to classify the 6 types of sensory neurons by size, type of receptors, level of myelination, and type of information transmitted. So the 6 types are listed there. The 1 alpha, 1 beta, 2 A beta, A delta, and C fibers. We talked about we met the A beta, A delta, and C's in the last quarter when we were talking about pain theory, and we will continue to talk about these guys. So the functional significance of axon diameter: larger axons transmit information faster. And so these 6 types of sensory neurons are listed by size. The 1 alphas are the biggest and the C's are the smallest. So the level of myelination, there's no specific measurement for that, but we're going to talk about more myelinated versus less myelinated. So more myelinated axons, more heavily myelinated axons transmit information faster. So you'll see some descriptions of axons that'll say they're unmyelinated, and really none of our peripheral axons are completely unmyelinated, but we'll just say they have a very low level of myelination. So I want you to be able to name the stimulus for the following sensory receptors: mechanoreceptors, chemoreceptors, thermoreceptors, and nociceptors. Nociceptors we met when we began to talk about pain theory. The other 3, you can completely guess what they encode: mechanical sensations, chemical sensations, and thermal sensations. So not too hard. Luckily, the name gives us the information that we need. So the sensory receptors, their job is to encode external information into receptor potentials. So we can then encode that information into something that the nervous system can read. Important, right? So sensory information from which receptors is used in proprioception to sense body position. And we'll talk about the different receptors, and you'll know the answer to that question. And where are the cell bodies of the primary somatosensory receptor neurons located? So the primary somatosensory receptor neurons are those pseudo-unipolar neurons where they have 2 axons and no true dendrites. So one of the axons has the receptor end and the other goes up to the central nervous system, and the primary and the cell body is in between those two, and we'll talk about its location. So somatosensation is really sensory information from the skin and musculoskeletal systems. Information from the skin can either be superficial or cutaneous. So superficial is like touch, pain, temperature. Cutaneous is like pressure, vibration, etc. And we'll talk about those individual bits of information that are cutaneous and superficial receptors are giving us. Information from the musculoskeletal system that is considered somatosensation includes proprioception and pain. So we have different sensors in the musculoskeletal system that we'll talk about that give us that information. So information in the somatosensory system proceeds from the receptor, which encodes that external information, through a series of neurons to the brain. So remember at the very beginning we were talking about the path where we're headed. We're headed to the cerebral cortex, and this is another brick in the yellow brick road to the cerebral cortex. Sorry, I watched The Wizard of Oz last night, so it's in my head. So cutaneous sensation, that sensation from the base of our skin, basically, the speed of information processing is determined by the diameter of the axons, the degree of myelination, and the number of synapses in the pathway. Sensory information from the skin is called cutaneous. Cutaneous means skin. So the diameter of the axons, if you have a larger axon, it transmits the information faster. If you have a more myelinated axon, it transmits the information faster. If you have more synapses, that slows it down because we have more areas where those neurotransmitters have to float across and activate receptors and develop chemical potentials and all that. So if you have fewer synapses, it's going to be faster. So just bear that in mind. Sensory receptors are specialized and they respond to only a specific type of stimulus. So if you have an adequate stimulus under normal conditions, that sensory receptor will encode into receptor potential, which will hopefully generate an action potential with an adequate stimulus. Mechanoreceptors are they are stimulated by mechanical deformation of the receptor by touch, pressure, stretch, or vibration. You should know those 4 things: touch, pressure, stretch, or vibration. Soft tissue mobilization, we're touching, we have light touch, we have pressure, we're stretching the tissues, and probably not vibration, but unless you're tapping or something. Chemoreceptors are stimulated by substances released by cells, including damaged cells after injury or infection. So they're also chemoreceptors. We talked about a couple of them in the autonomic nervous system, that the pressure receptors, those are mechanoreceptors that sense your blood pressure. The chemoreceptors that sense CO2 and everything. But the chemoreceptors in the somatosensory system, they are going to most of the time be sensing cells, substances released by cells like histamines, enkephalins, all that stuff that we've talked about before. So, or when you crush a cell, it releases a lot of chemicals, and the chemoreceptors detect that. Thermoreceptors, of course, transmit information regarding heat or cold. Sensory receptors that are located at the distal ends of peripheral nerves encode stimulation into receptor potentials. So The receptors are classified into two different classes, tonic or phasic. Tonic receptors respond as long as the stimulus is maintained. So the stretch receptors, and we'll talk about them specifically in a little bit, but a stretch receptor, as long as a stretch is maintained, you're encoding information and you're sending neural signals. Phasic receptors adapt to a constant stimulus and stop responding. This is the process of habituation that we talked about in chapter 7. So when you put on a watch, this is my example. When you put on a watch, right at first you feel it. Or when you put on your clothes in the morning, right at first you feel the clothes on your body. You feel that sensation. But later on in the day, you don't feel that anymore because those phasic receptors to touch have adapted to that constant stimulus, and they go, oh yeah, big deal, you have a sweater on. we don't need to know about that, or the cerebral cortex doesn't need to know about that. It's got more important things to deal with, and so we're just going to stop sending the signal, and we're habituated to that sensation. So the area of skin that's innervated by a single afferent neuron is called its receptive field. Receptive fields tend to be smaller distally and larger proximally. Distal regions of the body have a greater density of receptors than proximal areas. So this is the explanation for that 2-point discrimination that you did in Tests and Measures lab, if you recall that. If you have the caliper, if the two ends of the caliper are in the receptive field of the same neuron, so the example in the photo here, or in the diagram, is caliper A. Its two ends are in the receptive field of the blue sensory neuron, and so you sense it as one point. The B caliper, it has one leg in the blue sensory neuron and one leg in the receptive field of the yellow sensory neuron, so you sense it as 2 points. So if you have greater density of receptive fields, like in the fingertips, you can discern two very close points. If you're more proximal, like on the low back, it might only feel like 1 point because you have larger receptive fields. So the combination of small receptive fields and greater density of receptors distally enables us to distinguish between two closely applied stimuli on a fingertip. However, the same stimuli cannot be distinguished on the trunk. So I hope you experienced that in Tests and Measures lab because that's relevant for what we're talking about here. So the somatosensory first-order neurons, and we call them first-order neurons because they're the first neuron, they're the first step in the sensation experience. They are encoding that sensory information. They're the first line. So the somatosensory first-order neurons have two axons. It's that pseudo-unipolar neuron. The distal axon conducts messages from the receptor to the cell body. The proximal axon projects from the cell body into the spinal cord or the brainstem. So when we talk about projecting with neurons, it means that neuron carries information from one place to another. So if it projects to the brainstem, it carries information from where it is to the brainstem. If it projects to the spinal cord, it carries information from where it is to the spinal cord. So somatosensory first-order afferents are classified according to axon diameter. So 1A is the largest, the 1B is the second, 2 or A betas are about the same size, and then A deltas and C fibers are the smallest. So the larger diameter axons transmit information faster than smaller diameter axons. Larger diameter axons are more myelinated, and so they allow for saltatory conduction and they transmit information faster.